right, I noticed that there is a AirPod up here. Somebody's lunching this. I think it was up here on Wednesday though, so probably not anybody in here. Okay. Okay, so I don't have any big announcements today. The question that I keep getting from some of you is, hey, I answered this homework question right and I'm not getting credit for it. Remember, I have to go in and grade those by hand. I have to figure out some way to do that really kind of quickly. Otherwise, I'm going to have to start abandoning those questions because there's too many of them. I also have noticed that the amount of points that I assigned to each question is different than what I thought I assigned it. So I'm going to feel this out and see how it's going. I didn't intend for that homework to be taking you a lot of time. So, you know, if you have comments about the homework or complaints or whatever, it's working out fine, then I'd be glad to hear that for sure. I was thinking about canceling office hours for Tuesday. We're not running micro labs on Tuesday, and so I don't have any class that day. So if you were planning on coming to office hours, just let me know. I will probably be on campus anyway, but just maybe not as early as what my office hours are. So do let me know. By the way, did we win our football game last night? Oh, we did. I checked at one point, I saw we were ahead. All right, that's good. That's a big win. Good. Okay, well, let's continue where we left off. Last time we started to talk about culturing microorganisms, talked about how important that is so we can learn to characterize them and classify them. We're talking about different kinds of media. So media is the collection of nutrients that the microorganisms are going to use to grow. And remember, when we add Auger to the collection of nutrients, then that solidifies it. Okay, so remember, the bacteria don't eat the Auger, they eat kind of the other stuff that's in the medium, not the Auger itself. All right, so last time we talked about some all-purpose media and the kinds of things that, sorry, my handwriting's so sloppy there, I notice that now. Kind of why we put certain things in media. And then we have some other kind of categories of media that I want you to be familiar with. And at some point, you'll talk about some of these things in lecture as well. All medias are, sorry, you'll talk about in lab if you're in lab as well. I think I said lecture. All media is considered to be either defined or complex. Okay, a defined media, that just means that the exact chemical composition is known. So if you can see an example here, glucose salts agar. By the way, don't memorize anything about the examples. They're just illustrating the principle. This is a media that I'm not familiar with, but I found the recipe online here. You can see we've got chemical formulas here. We know the exact elements of everything and the exact amounts as well that's in there. Okay, so we don't very often make up a defined media unless we have to, because they're very cumbersome to prepare, a little bit more time consuming. So we don't normally do that. Sometimes we would do that if we're trying to grow up an organism that doesn't really need a lot, like a minimal medium. Maybe it doesn't need a lot of different special things for it to grow. More often, we're gonna use a complex media. As you would guess, the exact chemical composition is not known. It's more of just kind of a slew of things thrown in there. So basically, if you look at this tryptocase soy agar recipe here on the right, you'll notice there's a couple of different digests, casein digest, soybean digest, or if you see the term extract, like a soybean extract, that just means soy that's been chemically digested to release a slew of vitamins, minerals, nutrients. We don't know exactly how much of what, usually enough to support growth. Okay, so again, more often we're going to be using some complex media. Just look for things that again don't have the chemical formula in there. Media can also be considered selective, differential, both of those things, or neither one. Okay, if it's considered selective, that means it is going to contain ingredients that inhibit certain kinds of organisms. Okay, so the key word there is inhibit. There's something in that media that keeps certain things from growing. Sometimes it might be an antibiotic. In this example, Sabarod's agar. In this example, it looks like they've taken a soil sample, or what is often done is you take a sample of soil and add some water to it to make kind of a slurry and put like a thin film of it on top of an agar plate. So that's what this looks like right here. You get just a ton of different kinds of colonies, different kinds of growth, what you would see in the soil. If you would do that and add it to nutrient agar, which is not considered selective, and you get all sorts of things. Sabarod's agar is very similar to nutrient agar, except the pH is a little bit lower. So that actually favors the growth of fungal colonies. Okay? A lot of bacteria, there are bacteria that grow fine at pH 5, but a lot of fungi prefer that. So when you grow it, when you have the low pH, that inhibits a lot of bacteria. And what you see are these fuzzy colonies, some of them quite large, that are characteristic of fungi. Okay, so essentially the Sabarod's agar is selecting against bacteria because of that low pH. Now, Sabarod's agar could also be considered an enrichment culture. And that just means an enrichment culture means that it fosters the growth of a desired organism. Let me add to that, that is present in low numbers. Okay, so say you're looking for a particular kind of organism, the Sabarod's agar could be considered enrichment, because if you're looking for fungi, okay, there were fungi in this plate, but in very small amounts, so it got kind of out-competed. Okay, so if you kind of shift the conditions that are more conducive to that fungi, then the bacteria kind of get out-competed. All right, so again, something that's present there, but it's a media that gives certain kind of media a better chance of surviving. An enrichment culture may or may not actually kill other organisms. All right, and then a differential media is media that contains ingredients that causes some growth, or we'll say some organisms, to have a changed appearance. So in other words, everything's growing, but some organisms will look different than other organisms because of some particular biochemical characteristic they have. Okay, so they use a different example in the OpenStax textbook. They use this McConkey agar, which if you're in lab, you're going to use later in the semester. In McConkey agar, they give the organism's a sugar source, and then there's a pH indicator. Organisms that can use that sugar will have acidic end products, so they'll turn a certain color. Okay, so a lot of times a differential medium might include a pH indicator. Here's another example of differential media using blood agar, and that's another media that you'll use in lab later. But organisms have 3 different ways that we describe how they break down red blood cells. Okay, so they may have one of 3 different appearances on blood agar. They might have enzymes to totally lyse red blood cells. We call that beta hemolysis, and you can see how those red blood cells in the media, it's actually sheep's blood agar, is totally broken down, so it looks kind of yellow. You could actually read through that plate if you had organisms that were beta hemolytic. Some organisms only have enzymes to partially break down those red blood cells, so the red blood cells get lysed, but the hemoglobin is actually not broken down. So you get more of kind of a greenish, brownish color change there. And then some organisms can't break down red blood cells at all. So there's no change in the appearance of the media. Okay, so the idea is we can streak an organism out in that medium, and we know something about the organism based on its appearance on a differential media. Okay. And then the last term that I want you to be familiar with is an enriched media. So an enriched media contains things, contains growth factors. So a growth factor is usually something organic. It might be like an amino acid, for example, maybe a vitamin or other nutrient that's going to promote the growth of a fastidious organism. And a fastidious organism, I like to think of that as just picky. So an organism that has to have certain growth factors or else it's not going to grow. So an example of a enriched media is this chocolate agar. And it doesn't actually have chocolate in it, unfortunately. It looks kind of brownish because this hemoglobin solution that we have in it just kind of turns a little bit brown when we heat it up. But we use this kind of medium, for instance, if we're trying to grow an organism like Neisseria. Neisseria is a gram-negative cocci, so a round cell that's gram-negative that is particularly difficult to grow in the lab. We have to give it this special media. It has, certain growth factors in there that normally wouldn't be in things like all-purpose media. So again, an enriched media is kind of giving something extra to certain kind of hard to grow organisms. So that term is easily confused with enrichment media. So I pointed that out down here. Remember the difference between the two. Enrichment media is when you're trying to encourage growth of organisms that are there in really small amounts, and you just don't want it to get kind of outcompeted by the other organisms. An enriched media has something special in it, so a hard to grow organism can grow. Confusing because they kind of sound alike. All right, so let me give you some practice with that terminology. All right, so read through this, take a minute or two and kind of jot down what you think is the best answer here. And you can talk to other people or do it yourself, either way. Have you seen this? Okay, good. All right, so this is kind of a complicated, multi-step, process question. I probably wouldn't have needed to give you even the recipe there. I told you everything that you needed to know within the question. So the first thing I said is organisms that can ferment mannitol turn yellow. Okay, so that implies that organisms that cannot ferment mannitol are going to be some other color. All right, so you know from that it's differential. Okay, and then I said only organisms that can tolerate a high salt concentration will grow, meaning that high salt concentration kills a lot of organisms. All right, so you know that it's selective. So in fact here, 75 grams out of 1,000, this is a 7.5% sodium chloride. That's, physiological salt is somewhere between 0.5 and 1. So that's a pretty hypertonic medium there. That a lot of organisms aren't going to be able to survive that. All right, so you know that it's both selective and differential. And then it's going to be complex because it's got this extract in it. Okay, we don't know exactly what's in there. Slew of stuff. All right, so best answer here: selective, differential, and complex. All right, and then is this a solid or a liquid media? Right, because you see agar in there, then that's going to be solid. Okay, good. So I'll try to give you a little bit more practice there, just kind of discerning between those different labels of media before that first exam. All right, so to wrap up here, remember there's lots and lots of microorganisms that we know about. We know that they're there because we can, maybe sequence their DNA or something, but we haven't been able to grow them up in the lab. It's a complicated thing. I think I mentioned before, we think that we are only able to culture in the lab about 1% of the organisms that are in our body, which you would think we would be most familiar with those. But, because maybe they need other organisms to grow as well. That's what's so complicated about our gut. There's so many different kinds of bacteria in there that's, that we can't simulate that ecological situation there in the lab. So we're constantly learning. There are some bacteria, for instance, the bacterium that causes leprosy, Mycobacterium leprae. The only way that we are able to study that is by growing it in armadillos. Okay, we haven't found a way to grow that up on any media in the lab. Likewise, the organism that causes syphilis, Treponema pallidum, it only grows in rabbits. Okay, it definitely makes it more difficult to study. And of course, viruses have to grow in living cells. So most often we will grow them in some kind of cell culture. The old name for that was tissue culture. But we have some kind of layer of cells that's bathed in nutrients. It's a little bit more difficult to grow viruses because those cells can very easily become contaminated with bacteria. And then it boosts everything up. And So even more stringent aseptic technique. We usually will work with viruses under some kind of fume hood just to try to keep down the contamination. Okay. Any question? Yeah. That's because we don't know the exact composition of the beef extract. Right. So remember, it's either going to be complex or defined. It's certainly not defined because, right, we don't know the chemical formulas of all those things that were released by that digestion. Okay, good. Thanks for clarifying. All right, let's move on. Okay. So a little bit about microscopy, usually a fairly interesting topic. And if you're in lab, you're going to get to do a good bit of this. I try to avoid as much like discussion of physics as possible when I'm talking about microscopy, but there is a little bit that we have to kind of dip into when we're thinking about seeing ourselves under the microscope and things that we can manipulate so that we can actually see them. So we're going to talk about some principles first. The parts of the microscope, I'm mostly going to leave that for your lecture professor. Oh, and by the way, if your lecture professor didn't tell you, those lab manuals are in now. So maybe after class, go ahead and grab one because the bookstore is closed on Saturday, Sunday, and Monday. So just heads up there. I thought of that because we were making students label the parts of the microscope in the lab book this year, and that's different. But okay, and then we're gonna talk about some kinds of microscopy. In lab, we only use one kind, but there's a lot of different kinds out there, just so you have a little familiarity with that. And then I'm just going to talk very briefly about staining. Mostly, we'll leave that for you to do in lab. But, a fairly short chapter, even though it kind of looks like there's many parts there. Okay, so again, some overlap here between lab and lecture, but in order to see your cells under the microscope, three things that you gotta have. Okay, you gotta have magnification, and that's pretty obvious. That's the apparent increase in size. We have to make those cells look bigger than what they really are. And with a light microscope that we use in lab, kind of like this, we can magnify up to about 1,000 times. And to get that total magnification, you multiply the ocular lenses here by whichever objective lens you're using. And like I said, this is an example, the picture there of a bright field microscope. It's a compound scope. Remember, that means that there's more than one lens in there. So Robert Hooke, first compound microscope. Remember, the first microscope was just a single lens, Antony van Leeuwenhoek. Okay, so, whoops, light microscope. So about 1,000 times as our limit. We could manufacture lenses that would give us even better magnification than that. But after about 1,000 times, there's no point in doing that because we start to get limited by the physical properties of light. Okay, so we start to be limited then by how good of resolution we can get. Resolution will define as the ability to tell that two objects are separate, but they're distinct from each other. So for example, when we use a light microscope to look at the cell wall and the cell membrane, it just looks like it's the same thing. Okay, we can't get good enough resolution to see that they're actually separate structures. Okay, so the resolution of a light microscope is going to be about 0.2. Yeah, 0.2 micrometers. Always have to stop and think which syllable to emphasize there. 0.2 micrometers. So resolution is actually, it's a distance, okay, between two objects where you can still tell that they're two objects. Okay. So if we want to think about, well, how can we monkey with our scopes to get the best resolution that we can? We have to understand that resolution depends on a measure of the wavelength of light. We usually measure that in nanometers and divide that by the numerical aperture. And the numerical aperture, again, we have to just talk a little bit about physics here. We're going to define that as a measure of the angles of light accepted into a lens. And that will be more clear in a minute. I'm going to come back to what the numerical aperture is. But if we want to have really good resolution is a small number. Okay, because again, that's the distance between two things where you can still tell that they're separate. So if they're really close together and you can still tell that they're separate, that's good resolution. If they have to be really far apart, then your resolution is not as good. Okay, so we want a small number on this side, the left side of the equation. So for the fraction to be smaller, either we want the numerator to be smaller or the denominator to be larger. Okay, so but think about, you know, your basic math. 1/2 is going to be less than 2/2, right? So we get that numerator smaller. Or if the denominator is bigger, right? 1/5 is less than 1/4. All right, so think about that. So how could we get the wavelength of light shorter? Well, if you think back to the size of wavelengths, different kinds of light. Okay, visible light, somewhere between 400 nanometers and 700 nanometers. So white lights, a normal white light bulb is going to be, I don't know, somewhere a mixture of that, somewhere right in the middle probably. So if we're using a, visible light, maybe if we used a purple light bulb, we would get a little bit shorter wavelength here on the top, a numerator, and in theory that would give us a little bit better resolution. I don't know if we'd be able to tell the difference though if we switched out our light bulbs in lab. If we went shorter wavelengths, ultraviolet wavelengths, somewhere between 10 and 400 nanometers, so we're getting shorter and shorter. And by wavelength, remember, we mean crest to crest or trough to trough. That's a measure of one wavelength. We get even shorter here, X-rays or gamma rays, even shorter electrons. Electrons apparently move in sort of a wave-like pattern, sort of not. They kind of act as a particle sometimes too. But that kind of illustrates, hey, if we use an electron microscope, okay, our wavelength is going to be way shorter, and that is going to give us much better resolution. Okay, so shorter wavelength light we can use, the better. What about this numerical aperture here? Let me illustrate that here. I'm not actually in love with this diagram, so I'm going to draw part of it again, but this is from your OpenStax. So to demonstrate what that numerical aperture has to do with anything, when we say this is your kind of low power scanning scanning lowest objective on your microscope. And you're going to use that to look at your cells on the slide here. What this is illustrating here, when the light comes up through here and it changes medium, it goes from coming through the glass to the air, it refracts out in lots of different directions, bends. Okay, so here's an illustration of light bending. Like, you know this if you've ever gone fishing and you've stuck your fishing rod in the water, it looks like it's bending off in a weird direction. That's just kind of the properties of light as it enters a different medium, the water. Okay, so we want to minimize that bending of light. So what we're going to do, it says it down here, we're going to use an oil immersion lens and oil. Okay, so basically we're going to use this 100x lens that when we're trying to see our cells here, we put a drop of oil right on the cells, and that lens will go right down in that drop. So that's what's pictured here on the left. That lens is designed to look right through that oil. And in fact, in the book, it says, if it's not in the oil, you'll still be able to see your cells, but you can't see them as well. I would take issue with that. I would say I have never been able to find cells if that oil immersion lens is not right in the oil. What the oil does is minimizes that scattering of light. So you can see here in this picture how most of the light is kind of coming up into the lens now, and it's not bending out as much. Okay, so what that does, the oil actually increases the numerical aperture. Okay, so it collects more of those angles of light, okay, which gives us a smaller resolving distance or improved resolution. Either way you want to say that. Okay, so two things, I'll write this here somewhere. So to improve resolution, either you decrease or shorten maybe the wavelength, I'll write that out, wavelength of light, or you use oil and an oil immersion lens. So in lab, we almost never even try to look at our bacteria without using that oil immersion lens. Okay, it has the highest magnification, 100x. Multiply that by the 10x of the oculars, so we have about 1,000 times magnification, and we've got the best resolution that we can with a light microscope anyway. Okay. The third principle. Any questions before I go on? The third principle, we also need to have good contrast. Okay, I wrote this, I have room to write more on this other page. Okay, contrast is just the ability of cells to stand out from the background. So, I mean, you could have great resolution and great magnification, and if those cells look the same as the background, you are not going to be able to find them. So you really need all three of those things together. What can improve contrast? Sometimes if you dim the light a little bit on your bright field microscope, that can help a little bit. If the light's really high, it just bleaches everything out. But if you dim the light just right, sometimes you can see the edges of the cell enough that you can find them. Even better, stain the cells. Okay, so these two pictures, the one on they're both human cheek cells. Maybe you did this in Bio 120. You kind of scrape the inside of your cheek and rub it around in a drop of water. The one on the left is unstained. That would be very difficult to find under a power that was high enough to see. The one on the right, I don't know, they added some kind of stain or maybe a couple different stains so that you could see the edges of that. Incidentally, the little dots around here, those are bacteria. So it gives you a little idea of how bacteria are 10 times, maybe more like 100 times as a rule, smaller than eukaryotic cells. Okay. Now, unfortunately, when we stain the cells, it always kills them. Okay. So it's kind of fun to see them moving around when they're alive, but you know, if they're dead, at least you can see them. Okay, and there are kinds of microscopy that we can use that enable us to see the cells without staining them, so we can still view them when they're alive. Okay, so the kinds of microscopy that we're going to look kind of briefly at, just so you kind of get an idea of what they are. I just listed them in this slide, and they're listed going from kind of the worst resolution to the best. Okay, so even though bright field is the most common, that and phase contrast give us kind of the poorest resolution all the way up to best. So I already talked about light microscopy a little bit. I'm sorry, bright field microscopy. This is the most common. It's just, I suppose, the easiest to use, but and like I said, about 1,000 times magnification is about our limit here. 0.2 micrometers for the resolution. What does the image look like? Usually we have a light background, and then again, most of the time we stain our cells, so they are going to appear darker. It does depend what kind of staining procedure you use, though. Sometimes you want to stain the background and then the cells are unstained. So it might look a little bit different, but your background is not going to be black. It's not going to be gray like some other kinds of microscopy that we'll get to. And again, I just put this on here. Don't worry about the parts of the scope for lecture. You'll get more familiar with that if you're in lab. But just wanted to put that there. Let's see. Okay. All right. So we do have a couple of phase microscopes upstairs. They don't really ever get much use, but it is a common kind of microscopy. I'll show you a picture of what we see in a minute. The advantage here is you don't need to stain the cells in order to get good contrast. Okay, so the big advantage, if you don't need to stain it, so you can view living cells. And there is another kind of microscopy that I decided to skip this semester called dark field microscopy, where you don't need to stain cells either. But because that kind of microscopy is not really used very much anymore, I decided to skip it. Okay, so the main idea, how do we get away without staining our cells here? Well, it's due to, we just kind of manipulating the light a little bit. So when light rays are in phase, that means that the crests line up with the crests and the troughs line up with the troughs. Then that doesn't really give us any contrast. We can't really see anything. But if we have the light rays out of phase, well, it's where here the troughs are lining up with the crest, okay, that's going to give us a little bit of contrast. So how this works here, if you've got your bacteria or your specimen that you're looking at and you pass the light through it, that bacteria will knock the light that's hitting it a little bit out of phase. Here it says deviated by a quarter wavelength. And then there's going to be a phase plate somewhere in the microscope. I don't know exactly where that is, that knocks that light out even a little bit more. So basically now that the light that came through the bacteria is deviated by half a wavelength. So that's enough contrast to create so that you can actually see the edges of that image. All right, so here's just comparing bright field microscopy. These are unstained cells. It's supposed to be kind of the same cell, different methods of microscopy. These two here at the bottom are phase contrast. This Normarsky is a kind of phase contrast that is just a little fancier. It's using gradients of light. Don't exactly understand how that works. It gives us a little cleaner image. Normal phase contrast, like you see here on the left, often you get this kind of artificial halo effect here around the cell. But hey, you can definitely make out sort of the edges of that image. And so you can actually, again, see it without staining. You can see dark field here has it's a dark background. Again, don't worry about that. We're not, like I said, it's not really used a whole lot, so we're not going to worry about that. Okay, so then moving down and using shorter wavelengths of light. This is still considered light microscopy, but a fluorescent microscope is going to use ultraviolet light. Okay, so remember that's shorter wavelengths. So better resolution. And the other thing to know about fluorescent microscopy is that it uses, not surprisingly, fluorescent dyes. Okay, so you might think, wait, UV light, that's kind of bad to look into UV light, right? Yes, there is a plate so that when you look through the scope that UV light, you're not shining that in your eyes. But so here's an example of how powerful this kind of microscopy can be. On the left here, this is a light micrograph of a sputum sample. So sputum is that stuff that you cough up like deep from your lungs, not just a little bit of saliva. So this really doesn't look like much. It looks like some junk there on the left with bright field microscopy. But if you add a dye that is fluorescent and adheres to a particular component of a certain kind of bacteria that causes tuberculosis, Mycobacterium tuberculosis, then you shine it, you put it into a fluorescent microscope, only those cells that dye adheres to will show up. So this is actually one of the ways that tuberculosis is diagnosed, to see if somebody has these certain bacteria in their lungs. Okay, so a fluorescent micrograph is always going to have a dark background, and the cells that the dye is adhered to are going to be bright green or bright orange. Question? Yes, so do the dyes kill bacteria? Yes, it's the staining process, whether there's lots of the chemical dyes, usually oftentimes we're heating the cells as part of, sometimes just briefly, but as part of the staining process. So yes, we can always expect them to be dead. And we can actually tag those fluorescent dyes to antibodies to make them really super specific and stick to just about anything that we want. So that's a powerful technique. Kind of a step up from a fluorescent microscope is a confocal microscope. They use ultraviolet lasers and fluorescent dyes. To give, and then basically these are a step up in complexity. A computer generates a 3D image. They actually have one of these over in Kinchy Hall. I've not actually visited it. But this is let's see if I can open up this link. This is a link in your textbook as well. of what a confocal image will look like, and hopefully makes a little bit more sense what I mean when I say 3D image. It's kind of bright in here to see that very well. But you can basically, you know, do a whole like Iron Man thing where you put an image up and you can turn it around and look every which way, because the computer has actually taken lots of sections and put them together. So it's often thought of as kind of a bridge between a light microscopy and electron microscopy. Oh, good, I'm back. Okay. All right. Starting to get really expensive here. We thought about at Grand Valley getting an electron microscope for a long time, but we settled on the confocal. We decided that could do most of the things that researchers wanted the electron scope for. With an electron scope, a real jump in complexity. Oftentimes you need a technician to run it for you. Again, just getting even more and more expensive here. But because the wavelengths of electrons are much shorter than light, we get the best resolution. And there's a couple different kinds of electron scopes, but in general, the cells are going to be fixed and viewed in a vacuum. Oops. 2 U's. The reason is that electrons get scattered by air. So we have to prepare the cells in the absence of air and view them in the absence of air. Let's see. And we get really, really good resolution though, somewhere between 10,000 and 100,000 times magnification. They have the ability to magnify even up to 1,000,000 times. That's not done as commonly, but because we get good resolution, we can really get good magnification. Okay, so I got a couple more minutes here. Transmission. So there's transmission electron micrographs and electron micrographs, or scanning electron microscopes. How you tell the difference there, the transmission electron micrograph, when we prepare the cells, they're going to be sliced really super thin with an ultra microtome. So if you think, you know, you go to the deli and you see them slicing off slices of turkey or whatever, think that, only you're slicing many sections of a single cell with the ultra microtome. Okay. So the image that we see is going to be a cross section. And when you don't really know what you're looking at, that's not always immediately obvious, but both of these are cross sections. The one on the left has been artificially colored. You're not normally going to see any dyes or colors like that. So it's always going to be black and white. But the image is always a cross section. This gives us our best resolution, about 0.3 nanometers. Okay, so 3 to 4 powers better resolution than the light microscope. Okay, making up the specimen is quite laborious. Okay, you have to usually dehydrate the specimen, embed it in plastic so that it can be sliced really, really thinly. Okay. We are out of time, so we'll finish up with that next time and start our cell structures, chapter 3. Okay. Have a great Labor Day. We'll see you Wednesday.